Hello, welcome back. In this little short video we're going to talk about meiosis, which is the way a cell divides that produces cells which are different to itself. And any sexually reproducing species uses meiosis as a way of creating the gametes. In the males, spermatozoa; in the females, the oocytes or egg cells. So we have our basic cell, our germ cell, and we have our nucleus, just the same way as we had in mitosis. Now again, we're starting from chromatin, which is just the static-like appearance of the nucleus before the chromosomes become visible. So one of the first major differences between mitosis and meiosis is there are two divisions of the nucleus. So it goes through two stages, not just the one in mitosis. Now in the first division, although they follow the same prophase, metaphase, anaphase, telophase, there is a very subtle difference between the two which creates the variation. Whereas in mitosis you end up with identical, the slightly different arrangement on the metaphase plate. In meiosis, in the first go around, creates potential for variation. Alright, so let's just go through it quickly. So and this is, let's draw that a bit better. Meiosis I, this is the first nuclear division. We're going to see in prophase the same kind of thing. We're going to see the chromosomes appearing, remember, sister chromatids. Which are the identical copies, like a photocopy, of one side to the other. So on that chromosome, if you looked at it under the microscope, joined together in the middle there, here's gene 1, gene 2, gene 3, gene 4, 5, 6, 7, and so on. On the other side, there's 1, there's 2, there's 3, there's 4, there's 5, there's 6, there's 7. They're at the same. So the sister chromatids have the same gene, which is a sequence of DNA that codes for a protein, in the same place all the way along on that one chromosome. Remember, in the human, 46 chromosomes. Now, there's an important number to note with that 46 chromosomes, and that is half, 23 of the 46. So we have 26 chromosome pairs. And what that means is we have chromosome 1, 2, 3, 4, all the way down. So we have 2 copies of chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3, etc. all the way to 2 copies of chromosome 23. Now in mitosis that doesn't matter because all 46 just line up along the prophase, on the metaphase plate just after prophase. But it becomes important in meiosis because they arrange on the metaphase plate differently. So same thing. Prophase, we see the dissolution of the nuclear membrane, we see the appearance of the chromosomes, and we get the appearance of the centrioles, north and south. So, you know, prophase has gone pretty similarly to mitosis, all good so far. Right then. The big thing in meiosis comes next, metaphase 1. Remember, meiosis 1, metaphase 1. Or metaphase of meiosis 1, depending how you want to describe it. Now, in mitosis they went in a straight line. Now watch what happens. Alright, so here is our 8 chromosomes, but this time they'll look different because remember I just drew one line for the mitosis and said 8 chromosomes. There are actually 4 chromosomes but 2 copies of each. In the 8 here in meiosis, the start of meiosis, here's your 8 chromosomes. Now, chromosome 1, 2, 3, 4, 5, 6, 7 and 8. Now they don't line up in order 1 to 23 because they don't realise that they have numbers, but they line up along the metaphase plate in their homologous chromosome pairs. That's what you're seeing here. So chromosome 1, each copy sits directly, above or below each other. Chromosome 2, chromosome 3, 4, etc. all the way to 23. So they line up in their homologous chromosome pairs. Now that is important because, ping that cap across the room. That's important because, if I just take the numbers away for a second, there's our metaphase plate. Here come our kinetochores. So there we are, metaphase in meiosis 1. Similar but not exact to mitosis metaphase because of the different arrangement of the chromosomes, because they're lining up now in their homologous pairs. So when we then move on to anaphase, we see something very significant. Anaphase, let's do away with the middle section here because they're being pulled apart now. Let's see what happens. So there we have it. We now have a separation north and south. We've still got chromosomes 1 through 8, chromosomes 1 through 8 down to the bottom, but they're still in their sister chromatids. So what we've done is we've halved the chromosome number, the total, because this 8 going across with the pairs is an actual. In this particular example I'm showing you, the total is 16, but there's 8 chromosomes, but there's 2 copies of each chromosome. So normally in mitosis they'd have all lined up 16 along the whole thing and separated out by sister chromatids, and then you'd have 2 cells which would have identical DNA to the original. Now in this one they haven't lined up all together, instead they've separated by their homologous pairs. So now when this cell goes into telophase, and does that and separates by cytokinesis, the whole cell's separated. We're just really looking at what's happening in the nucleus right now, and that all reforms around it. We're now down to 1, 2, 3, 4, 5, 6, 7, 8 and 8 there, where there was 16. So we have half the chromosome number. That's the important thing. You half chromosome number during meiosis I because of the way you line up your chromosomes on the metaphase plate. You do it by homologous pairs, same chromosome pairs. So it's like, in a sense, if you had a party for twins, and so you've got 8 sets of twins in your room. Now they're going to do a dance. So you ask them to line up in their twin pairs. So twin pair number 1, twin number 2, 3, 4, and they all hold hands in the middle, all line up holding hands. And then you ask them one side of the twin pair all to go to the one side of the room, and the other side all go to the other side of the room, and you've separated them out. So you've got a representative of each one of the twins here, identical twins, and each one of the identical twins up here. So that's how you've separated them out. Whereas in mitosis, you still have your same twin party, but instead of them all lining up holding hands, you line them up like they're going to do the conga, one after another. And then, well, of course, this is where the analogy breaks down, because you're hardly going to rip them in half, north and south, because that would be absolutely and utterly horrible. So hopefully that weird analogy will help you get in mind what's happening here. So we've created 2 cells. The cells have just had their chromosome number halved. One other important thing has occurred during this process. If we just wind this back a little bit, and we go back to them lining up on the metaphase plate. Now I've got them a little far apart at the moment, but something else occurs when they line up like this, and that is that you can have synapses. So a little bit from here can be spread onto this side, so they can actually swap parts of their chromosomes over. It's still this part here, and if we drew the one next to it, so what we're showing here is synapses. And what we're doing is we're getting a slight change. We're getting So we're getting a crossover of the first 2 genes as the homologous pair come together. So this is chromosome 1, this is chromosome 1, and they've swapped over. So you're getting a slight change. Because although they have the proteins or the genes in the same position, they may code for slightly different proteins. There's a bit of variation. Now where did these copies come from? Well, originally they came from how you were made into a fertilized egg. 23 of your chromosomes came from the sperm, and 23 of the chromosomes came from the egg. And when they came together, they re-established the 46 chromosomes, but of course chromosome 1 to 23 from your father, chromosome 1 to 23 from your mother. So they have all of the same gene sites, except for a slight difference on chromosome 23 itself against the sperm and the egg, which we'll look at in reproduction. But they could be slightly different variants. So chromosome 1, gene 1 here could be for eye colour, for example, could be blue on this one, and could be brown in the other one, which is why we get variations. So we can actually swap that up a little bit and change what you got from your parents, so the child's grandparents over. So you get a slightly different mix of what you already have. So that's crossing over and synapsis. Another part of the variation that occurs here is that we don't know for chromosome 1, 2, 3, 4, 5, 6, 7 and 8, of the ones that go into this cell here, how many came from your father or your mother. So you could have one where all of these were from your mother, all of these from your father. Or you could have this one, this one, this one from your mother, this one from father, this one from mother, that one from mother, and then 6 and 8 came from your father. Because there's 23 of these, all independently assorting, you can have up to about 8 trillion, something like that. It's a huge, huge number of possible variations. So just by lining up on the metaphase plate during meiosis I, you can get variation by independently assorting whether your mother or father's chromosome 8, chromosome 7 lines on the top line and the bottom line, and then separates. You also get crossing over and synapsis, so swapping around of the genes anyway. So that's 2 possible variations, and you have the chromosome number. That's what meiosis I does. Alright, I'm going to come back and talk about meiosis II.